So do you want to introduce him and then he can go?
All right, so Leon is a student from Augsburg University, and he's going to be talking to us today about properties of an active self-propelled system through obstacles. Okay, so go ahead and take it away.
Hi, can everybody hear me okay? Is this a good volume?
Yes, we're good.
All right, perfect. Yeah, hi, like she said, my name is Leon Armbruster, and I will be telling you about the first passage properties of an active self-propelled system through obstacles today. Our motivation is quite simple. It comes from a principle called loop extrusion. Each DNA strand is about 6 feet in length, but needs to fit inside the nucleus of a cell, which is about 5 to 10 micrometers long. And it does that by forming loops and then condensing. So first it curls up and then it condenses to a smaller size. And this process is mediated by proteins like cohesion that walk diffusively along the DNA backbone. On the bottom left here, you can see a quick mean squares displacement plot versus time, with the slope of 1 indicating a purely diffusive property. To the right, you can see a quick illustration of how it would look like, a cohesion protein ring walking along the DNA backbone. And as you can see, DNA backbones aren't exactly empty things. They're proteins that can be in the way, for example, nucleosomes, that change the effective diffusion constant there. So on the right, you can see a quick plot of the permeability, with the stars indicating regions where you can find nucleosomes. So you can see whenever a protein encounters one of these nucleosomes, the permeability drops by quite a severe amount. So from this principle, there is a puzzle that we set out to answer. The time required to form a loop with this low effective diffusion constant is large compared to the loop formation time. And we get motivated to look at it by a certain way from a counterintuitive result from a simulation. On the bottom left here, you can see a quick illustration of the simulation, with that one-dimensional lattice being the DNA backbone, the nucleosomes being extended barriers of length d, the region between nucleosomes being called the linker length delta, and a little protein that hops with a hopping rate p in region delta. And when it encounters an obstacle, a nucleosome, it changes its behavior. So then it hops with a rate of q above it or repels. If we now look at the time required for this little protein or this little walker to hop a certain length along this lattice versus the linker length, so versus the space in between the obstacles, we see a pretty surprising result. You would think that the more obstacles there are in its way, the longer it would take, because it takes a longer time to pass the barrier. But that's not actually the case. We can see that there's a peak when the linker length is around 100, which in this experiment would correlate to the length d also. So when the nucleosome and the linker length is approximately the same length. So this is this non-monotonic behavior we'll try to set out to see. So this leads us to 2 questions. Can we engineer an experimental design principle to test this phenomenon? And second, is this phenomenon generic? And does it happen for any kind of obstacle? To answer the first question, yes, with a design involving geometrically patterned 2D obstacles and a self-propelled robotic bug. And to answer the second question is no, it turns out that the nature of obstacles does matter. And the keyword up here in the first question is the self-propelled robotic bug that sets out to mimic that protein walking diffusively along the DNA backbone. And here we have to first characterize the system. We have to make sure it is actually diffusive. So on the top left, you can see a quick illustration of the bug with a quarter for scale. And on the top right, you can see a sample trajectory. Yes, you can see it from these loops, again, staying more in one region and not traveling far out. And on the bottom, you can see a mean squared displacement plot versus time. And here are 2 distinct regions. The first region is about the first second of the bug moving in free space. The slope is about 2, which correlates to super diffusive motion. So it travels more towards one direction, which is what you can see in the trajectory on the top right. And then after that initial second, it moves purely diffusively, again, forming these circles around. And that's exactly what we wanted to see, behaving purely diffusive after about a second, which models the protein. And this leads us to our first design of the obstacles, the nucleosomes in our system. So what we did is we chose wooden pegs and represented nucleosomes. And we arranged them in sort of a patterned region. So a region without them, without obstacles, and then without with along. Again, important to remember that all we want these obstacles to do is change how diffusive the robotic bug moves through them. And from here on out, I will also refer to the region with obstacle as A and the region without obstacles as B. And here's a quick sample trajectory of a bug moving through the system and the time it takes for the bug to move from one end of the boundary to the other end for the first time will be called the first passage time. And then it follows that the average of all of these will be the mean first passage time of many trials. And on the bottom left here, you can see a quick illustration of 5 of these trajectories. And here I have a quick video for you to see. Again, it's sped up a bit just because it will take, it's quite long, but you can see that while it moves in the region with the wooden pegs, the behavior of the bug changes. I will just jump ahead real quick. You see it started on the right side, and then after about a minute, it will make it through all the way to the obstacles, and will record the first passage time. And this leads us to our results. For this design principle, we saw no non-monotonic behavior. So we had 2 different protocols we sort of did. With protocol 1, where we keep the width of A constant, but increase the number of obstacles, the number of A's. And for each of these setups, we take about 100 readings and then take the average of that. Here on the graph on the right, you can see the normalized mean first passage time on the y-axis and the ratio of A over B on the x-axis. And as you can see, it is increasing the entire time. Important to note is that at A over B about 1, you see a decrease in the rate of increase. So it kind of levels off afterwards. For protocol 2, we keep the number of obstacles constant, but we increase the width of obstacles, again, taking about 100 trials for each of these setups. You can see similar behavior. Up until A over B, approximately 1, you can see a steep increase, and afterwards it sort of levels off. And if you remember back to the one-dimensional simulation, that is not what they found. They found that non-monotonic behavior, that first increase and then the decrease. So we set up a second design with different barriers. So instead of having these wooden pegs, we now have solid obstacles with tunnels cut in between them to change the diffusivity, which you can see on the top left here, and on the top right, just a quick side view of these tunnels. On the bottom left, you can see 2 sample trajectories of how that robot behaves in these regions. As you can see, it moves a lot in the regions where there is an obstacle, and every now and then it passes through one of these tunnels. And once it passes through the last tunnel, it goes all the way to the end. And here we see the non-monotonic behavior. So again, for protocol 1, we increase the number of A's. And we see that at the ratio of A over B equal to 1 is a peak, and then afterwards a decrease, and beforehand an increase. And for protocol 2, we see that A, where we keep A, the number of A constant, but increase the width of A, we see the same behavior increase up until A over B equal to 1, and then a slight decrease. It is important to mention that that last point for protocol 1, we're not quite happy with. We ran into some experimental setup issues. It's just about when the ratio of A over B is too big, so the region of B where the bug can actually move around is too small, it can't behave normally anymore, meaning it's too small, it can't really turn around as well as it usually does. So we think that is why that last point is up too high. And we're currently still working on figuring out maybe taking some more data a little earlier or changing our design of it. And this leads me to some of the analytical solutions our collaborators did for us. On the top here, you can see for design 1, so our wooden pegs. On the bottom for design 2. And on the left here would be the varying number. So again, protocol 1, where we just increase the number of obstacles. And on the right, you can see the varying width. So having constant obstacles, but changing the width of them. And they found exactly the same thing we found. For design 1, they found increase up until a certain ratio of A over B, and it sort of levels off for both the varying number and varying width of obstacles. And for the impenetrable area, so the tunnels in this case, they see an increase up until A over B is a certain ratio, and then a slight decrease. And that's again exactly what we found. And this leads me to my conclusion. So we successfully implemented 2 design principles to mimic the diffusion of an active self-propelled system for different spatial pattern regions. We found non-trivial first passage time dependence that does depend on the kind of obstacles we have. And again, very important to mention that the experimental results, the simulation and the analytical calculations for a purely diffusive random walker all agree with one another. And I would also like to mention, even though we are motivated by that DNA loop extrusion process, there are many systems in the real world that have a sort of diffusively moving active system in the presence of obstacles. So we think our research is applicable to other areas and to other systems. And I would like to thank my advisor, Momita Dasgupta, a lot, and my lab partner, Kang Yang, who did a lot of the experiments with me, and our collaborators, Sogata Gua, Mithu Mitra, and Dibenju Das for doing some of the analytical calculations. And I would like the NASA Space Grant, Ergo, and AAUW for funding our work, and the organizers for inviting me to talk here. Thank you very much. And if you have any questions, yeah, feel free.
Questions?
One of my questions is regarding the scale of the robot. Is there any particular reason that you picked that about quarter size? Or would you have potential to go larger or smaller?
It would have been better to go smaller. At a certain point, it is hard to find robots that exhibit this behavior that are much smaller. Again, it's basically just the smallest motor you can buy with some batteries in like a plastic housing. We would like to go a little smaller. There's some other ones we've looked at, but definitely not bigger. Again, if you make them bigger, the obstacles have to be bigger. And then that first passage time would also always be larger, which, would take more time to actually run the experiments. So we just tried to find the smallest one we could at the time.
Does this robot basically go straight until it hits something, or does it turn on its own? Ever?
It does turn on its own. Again, for the first second of driving, so that would be about, I think we looked at it's about 11 centimeters. It travels, and then there's a significant curve, and then it sort of starts traveling in these loops.
Any other questions? All right, let's go ahead and thank our speaker.
Thank you.
Stop every turn.